Right now, the major problems we have are EHP and white feces. So the current top threats to both of the most cultured species in the world, in Baname and Monodon, well, it depends. Depends on your species. Some species are more susceptible to some diseases than others. And also depends in the country. Some countries are affected by some diseases, some others are not, depending also on your biosecurity guidelines. However, for both species, the top viral threat is white spot syndrome virus. For bacterial threats, we have HPND, which is caused by a plasmid that produces a toxin by several species of Vibrio. And now, in the top parasite is the EHP and the white feces syndrome. Those are our top threats. In last year, we published a study that was carried out in around 200 ponds around the coast of Thailand. This study was developed to identify the risk factors related to HPND in the ponds, which at the time, it was the major threat to Thai shrimp industry. So around 200 ponds were selected at random, ponds that had been suffering from early mortality syndrome, and around 20 samples per pond were taken. Some samples were sent for histological analysis, some samples were sent for PCR analysis, basing the studies on the pathopancreas of the shrimp, which is usually the main target organ for disease. So based on these findings, we separate them in 4 groups. The group number 1, 41 ponds were positive for APEM by histology or PCR, which means around 20% of the 200 ponds were actually HPND affected. On the second group, we separated the groups that had bacterial lesions and histology, but no HPND-related lesions, and were HPND negative by PCR detection. The proportion between the first group and the second group indicated that early mortality syndrome was not equal to HPND. That was the first finding. The second, we had a third group, we had no pathology or bacterial lesions. However, we had collapsed hepatopancreas tubules and epithelia that could be related to maybe poor feeding practices in the pond. And in the fourth group, we had normal histology, but however, they still had some early mortality. What we found out about this study is around only 30 ponds were what we catalog as early mortality syndrome, but not all the signs were correlated also to APEM. So we differentiate early mortality that can be by many causes and APEM disease that are by a selected type of causes. However, another finding came from this analysis, which was very surprising for us. You can see the yellow ponds were ponds that were positive for EHP. At the moment, we didn't think EHP was a source of concern, since the characterization in 2009, where we found out that EHP was present, but it was not a cause of mortality. So it was put aside as not a priority for the studies. However, after this finding, we found that 119 ponds out of almost 200 ponds were positive, which indicated a prevalence of over 60%. This study are from samples taken in 2013, which, if we think about it, it's been 6 years now since we took the samples, so maybe the prevalence is even higher. So what countries have reported outbreak? This is not just a Thailand problem, not just a China problem. It has been reported in Indonesia and Malaysia, Vietnam and India, and in Latin America, which makes it a global problem. We have to work together to find an answer to this. So if you're not familiar with EHP, what is EHP? EHP is a fungi-related parasite. Some people catalog it as a fungi, some people think it's a parasite, but it's kind of like a mix between both of them. It was characterized first in 2009 by a group of Thai researchers from our laboratory. The heavy infection related to this parasite is related to the slow growth in the shrimp, however, is not a cause of direct mortality. And the effect of this infection is more apparent in the second month after stocking in the grow-out pond. This is from one of our examples in the farm. You can see in number pond A01, where we had 100% of infected shrimp. The weight related to the age of the pond was very low. In ponds where the infection rate was lower, the growth was a little bit better, which shows the effect that we have usually in the grow-outs. So after these findings, we have been actively addressing this epidemic, working jointly with international research group that includes groups from the UK, groups from China, from Japan. We have worked to develop several detection and study models to understand how this parasite works and how can we stop it. This work, we studied the EHP transmission, was done by an Indian student doing their master's degree. He did a very good job with this. We wanted to understand how EHP is transmitted horizontally in the pond. So he developed a challenge where we put, if you see inside the cage, we put infected shrimp, and swimming around that cage, we put healthy shrimp. So after a few days, we analyzed by PCR, and the healthy shrimps were infected. If you think about it, and if you know why, what can come out of the infected shrimp that can go through the healthy shrimp? So it's the feces. The parasite comes through the feces of the infected shrimp, goes into the water, and is consumed by the healthy shrimp, and they get infected. So that is like a very quick way of transmission, apart from the cannibalism and other aspects. So with thinking about that, we developed a method. How can we purify, or how can we obtain this parasite to study in a laboratory level and understand how it works? First, we know that EHP target organ is a pathopancreas. So we collected the pathopancreas of heavily infected shrimp. We homogenize it, we make it like a puree. We strain it several times until we get like a very small pellet. And then during a centrifugation step, we obtain a band. You can see this white band is the EHP spores that we are able to obtain in a very condensed manner. So we take those spores, we wash them, we use a mixture of a 2% fluoxin B, which is a commercially available chemical. We incubate them for a short period of time, and then we visualize under the microscope. So what you can see in the picture are the spores with what we call the polar filament when it's inside the spore. Once it comes out, it's called the polar tube. So this polar tube is the main route of infection within the cell, which means is the tool that they will use to pass on. So if we can stop this process, then we might be able to control it. So this was our main focus. Now we can obtain active and live EHP spore. Now, how can we kill them once we get them live? So that was our next step. If you want to understand how they infect the cell, the shrimp consumes the spores that are in the water, in the feed, or in the pond. Once they get inside the shrimp, they will shoot this polar tube that I showed you in the picture. Once they pierce through the cell wall, or they come in the cell, they will inject the infective material, and the spore will start growing inside the cell. Once it completes like all the reproduction of the spores will come out of the cell, and they will start again infecting other healthy cells. So this cycle goes on, and some of the spores will go into the environment, some will stay in the host. So what guidelines did we think about controlling this disease? We wanted to focus on factors that could inhibit the spore activity that could be applied by the industry in the farms to control the spread of the disease. We wanted to use physical factors, for example, time and temperature, and common chemicals in shrimp farming. We focus on chemicals that were easily available, not expensive, easy to use, and safe for the people that were going to use them. We were worried also about the small-scale farmers, that sometimes they don't have the resources or the equipment to use more complicated or more dangerous methods. So we tried inhibition in vitro in the laboratory using formalin, 65% active calcium hypochlorite or 65% active chlorine, potassium permanganate and ethanol, which are easy to obtain, easy to use, and easy to manage. So our first physical control in vitro was the use of freezing temperatures. We put active EHP spores, and after freezing for a few hours, we measured the activity. And after freezing for at least 2 hours at minus 20 degrees, we got a total inhibition of the spore, which can be applied as a biosecurity control. For example, when freezing live feed that you're going to give to your shrimp or your broodstock. If you're worried about freezing, studies have been done in polychaete, where the total fatty acid have been measured after a short freezing period and long freezing period. In this case, they focus on arachidonic acid, which is a precursor of prostaglandin, which prostaglandin enhances fertility and sexual maturation in the broodstock. So there were not significant changes if you freeze for a period of time before feeding, which can be used by using like a normal minus 20 degrees freezer, or you can use a deep freezer if you want to, and this can be easily applied in the farm as a preventive measure. So as a chemical control in vitro with formalin, we didn't get that good results. We still had around 5% to 10% activity, which in the, if you think about it, there is still a lot of activity going on. We were looking into get total inhibition. So when we tried with at least 40 ppm calcium hypochlorite, for example, this brand is the one we use, but you can use another brand as long as it's around the same concentration. After using around 40 ppm for at least 15 minutes, we got 100% of inhibition, or even 24 hours. But when applying into the farm, you might have to optimize. You have to remember that this study was done with purified spores in direct contact. But in your farm, you might have the spores in the soil, in the water, or in other surfaces of the pond. So you might have to increase and you have to optimize depending on the load. So potassium permanganate, at least 15 ppm of potassium permanganate was necessary in the lab. But this chemical is a highly oxidative chemical, which activity will be related to the organic load in your pond. So you might have to calculate how much do you need to control this disease. In ethanol, at least 20% ethanol, but if you're using 70% ethanol as a common disinfection tool, is also okay. But this one is mostly to disinfect surfaces, equipment, and personnel. We do not recommend this to be used with live animals, as all of these are toxic for the shrimp. But we do recommend for them to be used when preparing your pond before stocking, or when cleaning your equipment, cleaning your personnel, and cleaning your surrounding area. We know that high pH is one of the triggers for spore germination, meaning that at low pH, we didn't get much activity. pH 7, a neutral pH, we had around 10%, but at higher pH, like pH 9, we had a really high percent of germination. So we can relate this to the pond conditions. If the pH in your pond during growing stages goes above 9, you might have a problem when the infection rate goes up, if the spores get like overactive. However, you can use also this information in your favor, meaning when you prepare your pond, you can use lime to treat the surface and increase the pH and trick the spore to germinate. So if the spore shoots the polar tube, it will no longer have a way to infect the healthy shrimp. So you fool it into germinating before time. So you can use that in your favor too. This information is available in a paper we published with the full protocol. We published it last year, but we are trying to make this information available to more people that might not be familiar with it. What about white feces? The usual signs of white feces is the white gut content, as we all have seen. The feces float on the surface of the pond. This problem usually happens when EHP is present. If you don't have EHP, you probably will not have white feces. But if you have white feces, it's because you have EHP. So they usually come hand in hand. However, EHP is not the cause of white feces, and it has already been proven in our laboratory, where we proved that EHP was a component cause. If you think about it like a puzzle, EHP is only one part of the puzzle. There is also other factors that at the moment we are still trying to find out. However, our work is mostly focused on EHP. This information is also available already. In this experiment, what they did is we infected shrimp with EHP and we let them grow for around a week, and we let them be there for quite some time, but white feces did not appear. So we know it's only a component, but it's not the cause. However, we have some ongoing research where, if you let me explain this to you, we took samples from ponds affected with white feces and ponds that had no problem with white feces. Each band is around 10 shrimp from the ponds. And by doing 16S analysis studies, we identified that the samples that came from ponds with white feces, if you see those orange and red lines, means bacteria. So the ponds and the samples, the white gut samples that we took, had a really high load of bacteria that we have been able to identify compared to the healthy ponds. So these bacteria, the candidates, we are analyzing them, and we have been using it in laboratory models by using oral injection, for example, of these selected bacteria, and we have been able to obtain white feces in the lab. I'm not sure why the other pictures don't show, but there's another 2 pictures of white feces too. Currently, this study is going on, so I cannot give you a lot of details on it, but we are hopefully getting closer to finding a more appropriate answer on why this problem is happening. So we focus on developing these guidelines because we want our information from the laboratory be easy to apply in the farm. We think that disease biocontrol is possible through the optimization of laboratory results. So we hope this information will be useful for you as a potential guideline. At least it's a starting point. So we hope this can be used here. And I would like to acknowledge my supervisors, including Dr. Kalaya. She was not able to come here today. Professor Dr. Tim Flegel, Dr. Onshuma, and the Centex Laboratory at Mahidol University, and the Aquatic Research team, Biotech NASDA. Also, I would like to thank MPEDA for their invitation and all of you for sitting through this presentation and being patient. You can send me an email if you have more questions. There is my contact information. And if just to throw out there, we do some work with also kind of like commercially applied research. We have been working with some companies, international companies, where they want to test against these diseases. We have been able to support them. Also to farmers, if you have any questions that we can help you with, we are happy to do so. So please feel free to contact us, and thank you very much.